Alright, well, hi. We're at proteins today, and proteins are really cool. Um, they're great. A couple administrative kind of things before we start. Your quiz today is all online because people are here, there, and everywhere. Everybody takes it online. We'll get done a few minutes early. You can take it then if you want to. You have until 11:55 tonight to take it, okay? It's open book, open notes. It is timed, so the idea is that, like, you probably don't have time to go through everything you've ever learned about biology, so, like, know some stuff. It needs to be your own work, so don't, like, have somebody else do it for you. But it's open book, okay? So far, so good? Homework, I'm going to actually put the scores in today. So a lot of you have seen those already. It was brought to my attention that Master Biology is scoring some of your things slightly oddly. I'll fix that. Yeah. Questions about that? Concerns? Great. On to today. So we saw this on Wednesday briefly. Proteins do pretty much everything under the sun. They can be enzymes, they can speed up chemical reactions, they can make it so they happen in a biological time, they can be antibodies, they can be, they can act as storage, they can act as transport, they carry messages, they respond to stimuli, they move your body, they provide structural support, right? They're everywhere, all over the place. And I'm pretty sure I used mostly animal examples, but just for the record, proteins show up in slime mold, and they show up in moss, and they show up in amoebas. Anything that's alive, you're expecting proteins. So far, so good? Okay. Super complex, all over the place, from, sorry, I need to check I'm recording because I'm worried about not. There we go. Show up all over the place. They do so many different things, but they're all made from the same 20 amino acids. So you have 20 choices. How you arrange those is up to you. But you only have 20 building block choices. So, like, for my little bead string here that I have going on, I have 3 choices of beads. I've got pink, yellow, and red. Hi, online people. Yep. I can arrange those in whatever order I want, but I only have those 3 choices. For proteins, you have 20 options for amino acids. You can arrange them in whatever order you want, but you've got 20 building blocks. When you're just in this long string, it's called a polypeptide. So an amino acid is also called just a peptide. One little unit is called peptide, an amino acid. You stick them together before you have a bunch of them, a polypeptide. That's just the string concept. Strung together in order. Once you actually fold it up in some cool way, that now this can go and like chew up starch or whatever it is it's doing, now it's a protein. Yeah? I know, right? I really like these beads. I've decided I like playing with beads. So far, so good? Alright. Words just to show this. I brought a stick today, I'm very excited. All amino acids, all of our 20 amino acids have the same basic structure for the backbone. They have this amino group, right? Got nitrogen there. They've got this carboxyl group. They've got carbon hanging out in the middle, holding them both together. And they've got the miscellaneous category. Okay? This acts as an acid, right? See that hydrogen there? Totally gonna go. It's gonna act as an acid. This is an amino, hence amino acid. Yeah? You saw what they did there? Okay. All the amino acids have this amine group, so this amino group, and this carboxyl group, which acts as an acid. All gonna look like that. Where they're different is this side chain, this thing up here. Okay? In my continued quest to, we'll leave these for later. They all look like that. Oh, I need a big screen. Sorry. This is all of them. Here they are a little bit bigger. These are the 20 you get to pick from. okay? And you'll notice that the side chains vary from really, really small, up there, glycine, top left. The side chain's just a hydrogen, right, like it's little. Where something over here, arginine, right over here, that thing's huge, right? The side chains can be very different. They can have different properties as well. So, all right, all of these ones up here are non-polar. They're all hydrophobic. This thing looks a little bit like, that steroid we saw the other day. Not exactly, a little bit. This thing does not like hanging out in water at all. So all these ones are hydrophobic. All these ones up here are hydrophilic. Do you see that OH group? That hydrogen would love to hydrogen bond with something, right? These are all hydrophilic. Over here, these ones are charged. This one's acidic. These ones are acidic. These ones are basic. They'd react with each other, right? They're all going to be different. And that is going to set some things up. So you've got your little polypeptide, right? Let's pretend that all these pink ones are hydrophobic. They don't like water, right? They don't mix well with water. And let me make some more here. Pink ones are hydrophobic. And let's say the yellow ones are super hydrophilic. The yellow ones love water. Nothing could make them happier. So yes. Okay. Scared of water, love water. You drop this in water, how is the thing going to be folded up? Yeah, what you said, and sorry, I'm going to repeat like what you said just because back to class. The pink's going to be in the middle probably, and the yellow, the hydrophobic's going to hide from water, and the hydrophilic is going to be outside, right? That concept makes sense? It's now going to be what the characteristics of these side groups, are going to change how this whole thing behaves. We're okay with that? Yeah. Yeah. It gets more, but that's the basic concept, right? Something that's acidic and something that's basic, like I think these things can ion bond with each other. They get interesting. But you have 20 different types. You'll notice just for convenience sake, they've got codes for them. So all of these amino acids have their name, but then they also have a little 3-letter code, and then a 1-letter code. The 3-letter code is pretty obvious. You're like, oh, yep, glycine, GLY, right? They also have a 1-letter code, like G, right up there, which makes sense. The problem is some words or some letters get used more than once. So, because we've already used A, over here, this one's D. Don't worry about memorizing that quite yet, at this point in your life. But when you see it, just don't freak out, okay? It's just very, very hard to write, I don't know, lysine over and over again, right? So you just want a super easy code. Feeling comfortable? These are your 20 choices. These are your question. Are these your 20 choices only if you're human or if you're any kind of organism? Any kind, right? Jellyfish don't have a different set of these. The way they're organized, that's going to be different. But the building blocks are going to be the same, absolutely the same. Everyone solid on that? All right. So, polypeptides, lots of peptides, you can call amino acid a peptide. They're linked together by covalent bonds, and it's going to make just a nice, long chain of these amino acids. It should look like the polymers you've seen so far, right? We've got, up here, all these purple things, these are all the backbones of amino acids. They all are identical. regardless of what's sticking off on the side chain, these are all identical. Okay? So we've got this one, this was the amine group, the acid group. They're stuck together, right? There's this OH here, there's hydrogen here, there's a dehydration reaction like we saw with other polymers, water leaves, and now they're stuck together again. And now this end, the free end, you can add something else to. You can just keep doing that. You can keep adding things on, adding things on, adding things on. Yeah? For reasons, this end that you add things onto is called the C end, the C terminus. Anybody have insight into why it's called the C terminus? We've got, ignore H's, who cares about H's? Any guess what this side's called? It's the N terminus? C terminus? We're okay with that picture? You can add stuff onto the C terminus. You don't get to add stuff onto this side. So if you're building a protein, you need to put stuff in order. You can't do it like if you have a string and you can add beads on both sides, you don't get to do that here. You get to add just one direction. You can only add onto the C terminus. Okay? All is well with that? Great. Then the amino acids being strung together, it's nice, but it doesn't mean anything until they're actually folded and doing something. So once a polypeptide is folded into some functional unit, then it's a protein. Some of these words are just for like online study people as they're going through. It's very beautiful here. This is a protein from the flu virus. It's just a structural form. This is one way to represent the shape. Okay. It does, right? It's so beautiful. Okay, so this protein from the flu virus is folded in a certain way, right? It just is. Human antibody proteins happen to be folded for this flu virus is folded like this. Any guess what's going to happen? They're going to stick, right? They're going to stick because this thing is the same shape. Well, it's like the opposite shape. It matches beautifully. That was on purpose, right? Like this antibody works because it can find this miserable little protein from the flu virus, right? If you had all the same stuff, you had every single same amino acid, but you had rearranged it in a different shape, it doesn't work. Okay? We're okay with the idea? It needs to be in the right shape, and then it does its thing. And yeah, antibodies are really cool. When you're building a protein from a starting from amino acids building up, you have 4 different levels of structure. Okay? And I realize that you've been in school long enough that when you hear teachers be like, there are 6 main imports, or there are 4 levels of this, you're like, oh, right. Yes, we're gonna talk about these 4, okay? Briefly going through them, then we'll see each of them individually. The primary structure just means the first level, so like primary school, most basic. Secondary, you can think of like secondary school, like high school, it's the second level of structure. Tertiary is the 3rd level, just a fancy way of saying 3. Quaternary is a fancy way of saying 4, it's the 4th level of structure. With me here? Okay. Primary structure of a protein is just the sequence of amino acids. So I'm really committed to these beads right now. The primary structure is just saying something like, okay, yellow, 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 pink, pink, pink. It's just the order of the amino acids in that polypeptide chain. All right? This, incidentally, is the information you get in DNA. So, question. Is DNA protein? Say no. Right? It's not. It's absolutely not protein. Oh my goodness. It's nucleic acid, okay? It's not a protein. But DNA holds the information for how to make, it holds the information for the primary sequence for protein. All of what makes you, you're like, oh, yes, me, that information in your DNA is information for how to make these strings of peptides, how to make a string of amino acids, okay? With me here? I mean, like, there's more to your soul, etc. But from a biological standpoint, that's what you got, okay? With me? Great. So primary structure is something like this, where we just have all these amino acids strung together. There are covalent bonds holding them. Nice long string polypeptide chain. Primary structure. First level structure. Questions about this? Sweet. Okay, so I have this string. How can I make it more complicated? Yeah, like do something interesting to it. Okay. The secondary level of structure, I need to show you something. Here. Just a sec. Doing this online as well. Here, looking at just the straight, boring amino acid that doesn't have any fancy, doesn't have a fancy side group on it yet. What? Nope, you've got nitrogen, hydrogen, then you've got oxygen here. Watch here. I'm gonna make this big. Okay, if you look at the secondary structure, you'll see that there's this oxygen and nitrogen with hydrogen. We're gonna look right here. This oxygen would love to hydrogen bond with something. It can't hydrogen bond with this hydrogen that's like right next to its stuck on. The shape doesn't. It can't bend that much. But this polypeptide is long. This hydrogen over here, it can bond. So if you've got a long peptide chain, you can just fold it back and forth and back and forth, and we're going to only fold it once. The oxygen on this side and the hydrogen on this side can hydrogen bond, oh good grief. Can hydrogen bond across like this. Does that make sense? This is just set up because of the amino acid backbone. It can do hydrogen bonds, right? So it can fold like this. You could have another level if you had more hands than I do. You can just keep going back and forth, forming this pleated structure. It's called a beta pleated sheet. It's because of this amino acid, this polypeptide backbone. It sets this up. It can form this sheet, or it can also coil. So as you coil this around, you can have hydrogen bond interactions between those coils. You can hold this together now in a 3D shape. With me here? Yeah? Okay. You can see that. And it's held together in a given polypeptide. So you've got this polypeptide. A given one can have some parts where it's coiling, some parts where it's beta pleated sheet, some parts where it's a little bit straight for a minute, and then continues. You get to have all sorts of things. Finally, well, not finally. Okay. Next, sorry, I got time. Next, you've got the tertiary structure. This is the third level of structure. This is the overall shape of the stuff. Okay? This one, its shape is driven by those side chains, by the R groups. So the things that stick onto it. The things that are sticking onto the amino acids. Briefly, my string thing broke. I need something that sticks better because they fall apart. Okay. Primary structure, right? Secondary structure, we've got some sort of coils here. Oh my goodness. Problem. tertiary structure is here when, yeah, so we've got some coils over here, it's pleated over there, whatever. Now, because say these are positively charged, these are negatively charged, they're going to fold in a certain way. Okay? We're there? Lots of different things that can make this happen. You can have, well, here, I'll show you pictures. You can have something like this is an R group, this is a side chain, this is a side chain. These ones can hydrogen bond. The whole thing's the amino acid backbone part that can hydrogen bond itself, but these side groups can hydrogen bond with each other, right? Those can be forming some interactions. You can have hydrophobic interactions. So both of these side chains really hate water, so they glom on together so they don't have to hang out with water so much. You've got a negative charge here, positive charge here. These form an ionic bond, right? And now they're holding together. If you've got sulfur in the stuff, which you do, you'll form a disulfide bridge. This ends up being like a covalent bond holding it together. You can have lots of different interactions holding it together in different shapes. Comfortable with that? What's interesting, amongst many things, I'm sure this whole class is interesting to you. What's interesting to me personally is while you can tell based on, like, when you see a protein folded, you can be like, oh yeah, of course it's folded that way. Obviously, if we've got these things forming ionic bonds here and here, right, that's a reasonable way to fold it. However, if you just look at the structure cold, it's really hard to predict exactly which way it's going to fold, because maybe instead it's the ionic interactions folded this way, right? Like, it makes sense once you know how it's folding, you can go back and look why, but it's really hard to predict. If you ever can predict this, you will win the Nobel Prize. Like, I'm serious. There's a whole field of biology looking at this, of like, how can we figure out how it folds up, right? Because it's pretty easy in the lab to string amino acids together. You got 20 options. It's not exactly like stringing beads on, but you can be like this. You can make the string. But if you can get it to fold up right so it does something, that's really cool. Questions? Okay, so we've got tertiary structure. Tertiary structure is, for most proteins, that's where they're at. Tertiary structure, they've had their primary structure, that string of amino acids, covalently bound together. Secondary structure, it forms alpha helices. So one helix, multiple helices, just for words, or that beta pleated sheet. Tertiary structure, that whole thing then's folded in some pattern. Quaternary structures is just, for this thing to be a functional biological something, you need more than one polypeptide being involved. So collagen, this stuff is great. It's tough, it acts as a structural support. It shows up like fingernails, okay? Collagen, its quaternary structure, it needs 3 different polypeptides to come together. They're color-coded here. So this blue part, there's a bunch of amino acids in a string, and they're coiled in an alpha helice, okay? And then that alpha helice is coiled in this kind of bigger slow coil. Do you see that? That's it. This bigger slow coil is its tertiary structure. Its quaternary structure is when you stuff 3 of these things together, and it's basically a rope. Do you see that? Hemoglobin has a similar pattern. You need both the alpha subunit and the beta subunit. You need multiple polypeptides to come together. But you can see hemoglobin is a cool molecule. A cool protein. You can see there's some parts where we don't really see the amino acids here. They're just strung together. It's what's making the whole string. You can see some parts where it's coiled, right? Secondary structure. The tertiary structure is where the whole blue thing is folded. Blue or purple, by the way. I try really hard with colors, but I have been told I don't pick the same colors as you would pick always. Blue or purple? Blue? Purple? Both? Okay. I don't know. It causes me stress. My husband claims that he is going to teach our children how their colors because apparently, anyways. Okay, so hemoglobin, it's got two of these coming together to form the actual functional unit. Brief aside with hemoglobin, this stuff is amazing, and its shape's really cool. Hemoglobin carries oxygen, right? What's interesting to me about this is it's got places to carry quite a bit of oxygen, all right? Oxygen coming on changes its shape, so if it's already holding oxygen, it's easier to hold more oxygen. If it doesn't have oxygen, it's harder for it to hold oxygen. So if you have this stuff up next to your lungs where there's lots of oxygen, as soon as one oxygen comes on, it's easier to fill all the other spots with oxygen. Now the thing's full of oxygen, right? But if you're down someplace like in your big toe, where there's not such a high concentration of oxygen, if one oxygen leaves, it's easier for the other ones to all dump off too, because of the shape change. So this protein is amazing at picking up a bunch of oxygen and then dumping it. It's great. But the point of this is, quaternary structure is just you need at least two polypeptides coming together to form your functional protein. Not all proteins have to have quaternary structure. For them to be functional, all proteins have to have tertiary structure. Everyone's there? Questions? Things that could be confusing? Okay. I asked the 9 o'clock class if anybody had ever touched like a strong acid, and one person in that class had accidentally gotten phone battery acid all over her once. I know, right? Whew. If you want to share, anybody here ever touch a strong acid? Yeah, okay. Taking a person from last class, it didn't go well for her, okay? She was saying how it burned her hand. And she was saying it didn't burn her hand because it was hot. It wasn't hot like boiling water was hot. Like, the acid was just room temperature, but it burned. Can we all imagine that and be like, ooh, right? Okay. What's going on there is proteins respond to their environment. So it's fairly hard to break, biologically speaking, like if you're motivated, you can get that. But biologically speaking, it's fairly hard to break the covalent bonds that are holding together the primary structure of amino acid, okay? That's not what we're worried about. What we're worried about is you've got this nice 3D folded shape, the tertiary structure, that it's fairly easy to break, or you can break it. So the 3D folded shape that the protein's supposed to be in to do its job is called its native conformation. It's natural state, okay? If you bust that up, you're denaturing the protein. It's denatured, okay, outside of its natural state. So here, this tertiary structure is being held together by all these interactions, right? Great. For instance, there's an ionic bond here. If you say threw a bunch of salt in on this might not feel the need to bond with itself because it's got other stuff to do it with, right? Or say you've got some hydrogen bonds going on up here, but if you throw in some acid or some base, forget that. We're going to interact with the acid or base, right? So if you do that, you can mess up this 3D conformation, right? Because now it's going to unfold and break or be in a different way. Does that idea make sense? I'm assuming you burned yourself with heat at some point in your life, right? Yeah, it hurts, right? You physically break proteins. So adding heat to this can add enough energy that things jiggle apart the wrong way, right? In less painful ways, you do this with like eggs, right? So if you're eating fertilized eggs, I guess, this egg could go and become a chicken at some point. But once you cook it, you add energy to those proteins. You basically, you change their shape. You denature them. They're folded a certain way. You change their shape. And once you've done that, once you've broken these proteins, you're not going back to chicken, right? Like you've seriously you've denatured this protein. You've changed its function because you've changed its shape. So heat's good at doing this. pH, so how acidic or basic it is, can do this. This is why your body is very invested in buffers to keep you in the range that your proteins don't break. Temperature is good. So temperature, salt, pH are the big ones. Some other things can mess around with this too. Denatured proteins don't do their job because they're not the right shape to do their job. So far, so good? Want to see something creepy? Kind of? Okay, half the class says yes. So, this is a sheep brain. I realize you might not be like physicians or vets. Anything wrong with it? There's holes. You're like, oh, I might not be a vet, but you know, it's not good. It's not supposed to happen. So, mad cow disease is what happens to our little sheep buddy here. I know. In mad cow disease, the classic thing that happens is it's a prion disease. Basically, proteins go around getting unfolded. Just like, why not? And when you have a bunch of proteins unfolded in your brain, you end up with holes in the brain, and that's the technical term is bad. Okay, right? Like this is not good. A lot of diseases are actually associated with proteins misfolding or not being the right shape. Mad cow's disease, Alzheimer's, Parkinson's, they're all associated with the protein's not the shape it's supposed to be. Okay? We're okay with the idea? In high school, did you ever go to or middle school, did you ever go to a dance that had a chaperone? Yeah. Okay, so not what they were actually doing, but what were they supposed to be doing? Watching, right? Somehow helping guide you towards truth and light? Yes? Maybe? Officially? Okay, right? I think that's what they're supposed to be for, right? I don't think they're there just to eat the M&Ms. Are they? I don't know. Okay, officially they're supposed to be guiding you. Okay, yeah. Theoretically, we're going forward. They're supposed to guide you, okay? Yeah, no, I don't have so many analogies and I run out with them really fast. You've got your little polypeptide. It could fold up lots of different ways, but you need it to fold a specific way. All right? There are proteins that assist this. They're called, this is why my dumb analogy is happening, they're called chaperonins. I kid you not.
No, I don't.
Yeah, they're called chaperonins. And the idea of the chaperonin is it sees the polypeptide and helps guides it towards the proper shape. Okay, it helps fold it up the way it's supposed to be folded. All right? When the polypeptide is appropriately folded, it's got the interactions, it now can do its thing. Everyone's comfortable with this? We can go through primary structure. Oh my goodness, I keep breaking my little protein. Primary structure, amino acids in a chain. Secondary structure, coiled or pleated, right? That's set up by the amino acid, by the backbone. Tertiary structure is set up by the R groups or the side chains interacting with each other. And quaternary structures, two or more of these coming together. Yeah? Great. Last thing, I want to let you out 10 minutes early. We're not quite done yet. I want to let you out 10 minutes early so you can take your quiz officially on time. You have until this evening to do so. You don't need to push people off the computers nearby to get it done. But I do want to do the "Are you here?" thing, and I totally forgot to do it for the 9 o'clock class. I had to take physical attendance. It was very sad.
So when you're exposed to mad cow disease, is that why they don't let you give blood? Because
There were.
My mom was exposed.
Uh-huh.
She lived in England in the 80s, and she tried to give blood the other day, and her
Nope. Nope. So it's probably fine. Just sometimes it like, yeah.
Yeah.
And
It normally only affects humans.
right, but can it? So there's a version that's also.
Well, I know we're married, but I mean like farm animals.
Uh-huh.
It only really affects those.
Also, don't eat people. Like I know you weren't planning on it, but if you eat like human brains, you're way more likely to get this. So don't do that.
No,
I mean, for other reasons too. Say what?
I said, what about the rest of the body?
That's not such a big problem. I mean, it's a big problem for moral reasons, but for
Just don't eat the brain.
but for it's called CDJ. It's the human version of mad cow disease. Yeah. Anyway, so, but just for the record, class, don't eat humans for a variety of reasons. Yes, you guys need things.
I mean.
Gotcha. Let me get everybody and then hand them out to you.
But like, yes.
Sorry. Okay. Come on. Alright, thanks everybody. And yeah, people in the back, let me get you yours. Come on. Good heavens. I'll get them in your purple bubble.
You still didn't get me. Jeez, just take it. Just take it.
Whatever. Come on.
You're not holding it.
No, like all options should be available. There we go.
Nope.
No, you didn't take it?
There it is.
Okay.
There it is. I'm like, this freaking thing is. I'm here. I know you said bring it, but I'm one of those people that forgot it.
Okay. No, you're good. Sorry, it's just
I know where
that's all. Okay, that's all we've got for today's class. So if you want to leave, feel free to leave. Have a nice. It didn't take yours. Okay, briefly, everybody who didn't take, if you don't have one, I'll give it to you. Everybody who wasn't here, can you shout your name loudly?
Wow.
Still wasn't that loud.
That's okay. You're close. Zach and Adam. Do you guys each have them? Do you have them, or do you need a new one? You want to give them? Okay. Zach, I gave yours to one of your buddies who said he'd give it to you, but apparently he didn't. Oh, okay. He said he lives in the same dorm as you or something.
It was an African biology thing.
I was gonna say something.
I was the one who emailed you, like,
Uh-huh.
all of my stuff. It came in all at once. All the assignments finally popped up. I've been going day by day, and I only completed, like, the one.
Okay.
It all finally popped up
Okay.
out of nowhere. So,
Okay.
I have all of those to do.
You so not to panic. I mean,
Yeah.
Master Biology, she's gonna show you that things are late.
Right.
These first 3 weeks, just get them done.
Yeah.
Starting this coming week,
Yeah.
that one I will start taking points off for being late, but these ones just.
Yeah, I was gonna say they finally popped up.
Good.
So.
People have been having weird, like,
They
it's
have,
not personal,
like,
it's been
some
weirder
people
than it
talk
ever has
about
been
it.
before.
I'm like, what do you mean all these assignments? I go, there's nothing in there. They go, yes, there is. And they finally popped up a couple days ago, and I'm like,
Okay.
what are they?
Okay, so they're there. Good luck. I'm sorry that
Right.
your week is gonna get busier. Don't panic.
No, no, no.
I'm gonna upload grades, so it might look bad because you might be missing some stuff
Yeah,
yet.
I was looking in there, I'm like,
Don't
oh,
panic.
okay.
I'll keep uploading it
Alright.
as you do stuff.
Alright.
So yeah, good luck. And you don't need to feel like you need to do all the things before you do the next homework. You've got this. You're good. You're good. Yes. Oh, I feel so much love. Okay. Nice. You've got time.
I'm having problems with my outlet and my email right now. Like, I can't access them, so if I don't.
Just email me. Yeah, if you don't. Yeah, and like if your outlet turns on like 11:54, don't be like I have to do it in 1 minute. Like just it'll be fine. Yeah, but keep me emailed because I forget stuff.
Yeah.
Oh,
I
so-
didn't like register that I was here,
Oh,
so I just came to tell you that.
thank you. Hold 2 seconds. Stop recording.